Good day, this is Professor Will, CBMG PhD. Today's Monday, March 7th, 2022. It is 1:12 and 35 seconds, a.m. Eastern Standard Time. Today's research, bacterial vaginosis. So let's hop to it here. Researchers identify potential approaches to modify the vaginal microbiome. Insights may lead to more effective treatments for bacterial vaginosis and improve women's health outcomes globally. Date March 3rd, 2022. Source, Massachusetts General Hospital. The study shows that L. innards has unique nutritional requirements that distinguish it from L. crispatus, potentially allowing it to be targeted using novel therapeutic strategies. The female genital tract is naturally colonized by mixed communities of bacteria known as a vaginal microbiome. When these communities are dominated by species such as Lactis bacillus, crispatus, they provide important protective functions in genital health. But overgrowth of certain other bacterial species is linked to a condition known as bacterial vaginosis. Bacterial vaginosis affects nearly 30% of women around the world, carrying increased risk for sexually transmitted diseases, HIV, and in pregnant individuals, premature birth. Unfortunately, current antibiotic-based treatments for bacterial vaginosis are poorly effective, with high rates of reoccurrence. Lactis bacillus innards is the most abundant and common vaginal bacterial species worldwide, but it is poorly studied because scientists have had difficulty going into the laboratory under conditions used to culture species like Lactis bacillus crispatus. Researchers found that adding the amino acid cysteine to standard lactobacillus culture media allowed them to grow Lactis bacillus inner strains from samples collected from U.S. and South African women. And surprisingly, when the researchers analyzed a novel collection of more than 1,200 vaginal lactobacillus genomes from more than 300 women across 4 continents, they found that none of the species were able to make their own cysteine. The researchers therefore hypothesized that all vaginal lactobacillus species require external cysteine sources. They measured cysteine concentrations in vaginal fluid samples from South African women with high rates of bacterial vaginosis, finding that higher vaginal cysteine levels were linked to lactobacillus dominant microbiomes, while bacterial vaginosis was associated with low cysteine levels. The results suggested all vaginal lactobacilli acquire cysteine from their environment, but lactobacillus innards' ability to do so was more limited than other species. Indeed, when researchers looked at the genomes, they saw that all species, except lactobacillus innards, had multiple systems that are predicted to transport cysteine, or its oxidized form, cysteine. The team therefore tested effective compounds known to inhibit cysteine uptake, finding that cysteine uptake inhibitors selectively block growth of lactobacillus innards in the laboratory, but not other lactobacillus species. These findings were exciting because they suggested a way to improve bacterial vaginosis treatment by blocking lactobacillus innards growth in favor of more health-associated species like Lactobacillus crispatus. Researchers constructed mixed bacterial communities, including Lactobacillus innards, Lactobacillus crispatus, and various bacterial vaginosis associated bacteria in the laboratory. They then treated the communities with an antibiotic commonly used for bacterial vaginosis therapy with a cysteine uptake inhibitor, or a combination of the two. And their results show that the combination allowed Lactobacillus crispatus to outcompete other species more effectively than the antibiotic alone. The researchers believe these results suggest a path of better therapies. One reason it has been difficult to develop effective bacterial vaginosis treatments is that researchers haven't had the correct tools to study the vaginal microbiome in the laboratory. And here developing the right tools to cultivate Lactobacillus inners in the laboratory, immediately translated into an important finding that will hopefully lead to improved therapies for bacterial vaginosis. The team emphasizes that several important questions remain. It isn't yet clear how Lactobacillus inners take subsisting from its environment, and more potent versions of the inhibitors may need to be developed before the strategy can be used to treat patients. Even so, the study is a promising step forward for this common but difficult to treat condition. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.